Okay, in this section, we are going to be talking about bacteria and the differences between prokaryotes and specifically the domain bacteria and then archaea. So, bacteria are one of the most studied organisms on the planet. They're found everywhere. They have strong cell walls that are made of the peptidoglycan, which is the alternating sugar molecules. They also can have a second cell wall. Another prokaryote is archaea, and archaea are found in the most extreme environments on the planet. So there's thermoacidophiles, which thermoacidophiles, so meaning I love, phile means I love. I love boiling acid. So they love boiling acid. There's halophiles, which means I love salt, so they live in the saltiest environment. There's even methanogens, which can live with no oxygen whatsoever, like in sewage treatment plants, swamps, bogs, near volcano vents. They also are responsible for the lovely gas that is made in your lower digestive tract. And if you've had embarrassing moments because of that, you can blame archaea that can live there with no oxygen. So what's the difference between bacteria and archaea? So some differences are that the bacteria have cell walls that, and this is right here that you can reference that, have cell walls that contain peptidoglycan, but archaea do not. They have different lipids in their membranes. They have different ribosomal proteins and RNA. And the ribosomal proteins of archaea are actually more like a eukaryote. So there's some interesting differences. So the chromosomes of prokaryotes have a nucleoid, which is it's not a nucleus, but it's sort of like their DNA is in this long squiggly strand. I'm looking at this diagram of a bacteria. There's the pili, and the pili on the outside really help the bacteria to attach to surfaces. It can serve as a bridge between two prokaryotes where they can connect together and then share genetic information. We'll talk about that in a second. And then on the outside, they have a capsule. This prevents them from drying out. It serves as protection and it helps attach to things. Then there's like the flagella that helps it spin around and move. And then here are some basic characteristics of prokaryotes, and one is their shape. You can have a very round shape prokaryote. These are the cocci shape, which are circular. Then the bacilli, which are sort of like little round rods, and then this squiggly worm-like looking one are called spirilla. Okay, so those are the different shapes of those two, or those three. So bacteria can reproduce two different ways. One way is just by binary fission, meaning one bacteria duplicates its chromosome, splits in half. You get an identical copy of the next one. The other method is through conjugation, where they join together and then they swap genetic information across a bridge. So one takes in new genetic information and pushes out some of its genetic information, and they swap it. So why would two bacteria ever just conjugate when they can just make identical clones of themselves? Obviously, conjugation actually helps increase genetic diversity. So I want to go back to this binary fission, splitting in half. And bacteria specifically can do this once every 20 minutes, make an exact copy of itself. That's crazy. Make an exact copy of yourself every 20 minutes. So I want to show you what that would look like. If you start at 1 o'clock, say, and you have one bacteria and you multiply it by 2, at the end of 20 minutes, now you have 2 bacteria. So now the time is 1:20, and you have 2 bacteria and you multiply it by 2, and now you have 4 bacteria. And at 1:40, You take those 4 bacteria and you multiply it by 2, and now you have 8 bacteria. And at 2 o'clock, 20 minutes later, you take those right here, 8 bacteria, and you multiply it by 2, and what would you have? And you can keep figuring that out, but soon you get to some very, very large numbers of prokaryotes and how they are replicating. Now, prokaryotes can eat in a number of different ways. They can, heterotrophs, eat other things. Phototrophs use light for food to make energy like photosynthesis, or chemoautotrophs, where they're making energy from chemicals that are near them. So how do bacteria survive when things get really rough? Well, the interesting thing here is they actually survive really well. One of the things that they can do is they can throw out what's called an endospore, where it kind of reminds me of, like, in Harry Potter, Voldemort threw out chunks of his heart to different places and hid them all over, so that he couldn't really die. This is what some bacteria can kind of do. It reminds me of an endospore. What it does is it takes basically a small bit of their genetic information and can make this little endospore, which is like a seed, and it's very well protected, so that it really can survive a harsh situation. So even though the bacteria has died, the endospore with just the genetic information will go on and survive. And then when conditions get better, it will sprout and germinate again to build a whole new bacteria, which is just crazy that it can do it. Another way that bacteria can survive is that it can make mutations, make copies of itself. So the mutations, and there's no picture of this, but just because bacteria can make copies of itself every 20 minutes, it ends up having a lot of mutations, because that reproduction process is happening really often. Whereas humans might take 20 years to reproduce or something, a bacteria can be once every 20 minutes. So there's just going to be more mutations in general with a bacteria. So then bacteria have their whole own ecology. So bacteria are amazing because they can help us cycle nutrients in the ecosystem, like nitrogen fixation, where they actually take nitrogen and put it in a form that we can use it. And they help us digest nutrients from food. We use them to make foods like yogurt and some medicines. We do all kinds of different things with bacteria. So one of the cool things that, like I just mentioned, is here's normal E. coli. And E. coli in your intestines is totally important for you, to help you out. Now, if you have E. coli ends up not in your intestine, in the other places, like if there's like food poisoning and you end up eating some E. coli in your upper digestive system, that's a major problem. But in your lower digestive tract, this is really an important way that you gain your nutrients. In fact, some things like vitamin K is made because of E. coli breaking down your foods. And, I would encourage you to go look up some videos on how bacteria really, they've documented bacteria are sending messages up to our brain to make us crave certain foods that they digest, which makes me think it's really important for us to be careful with what you're eating to make sure that you're eating healthy things. So here are different human bacterial diseases that you can look up that are caused by bacteria, and I would encourage you to look at that too as you continue your study of bacteria.